Are you a podcaster? Maybe you've got that big idea and you're looking for a network to join. The multi-award-winning Ozcast Network can get your content to eyes and ears all over the world. Join now for the first month free, and you could be featuring this sound at the beginning of your podcast.
Ozcast.
Simply head to ozcastnetwork.com for details.
Welcome to This Medical Life podcast. These are the stories of medicine with Steve Davis and Dr. Travis Brown. This is the story of familial adenomatous polyposis. Dr. Travis Brown, for the remainder of this episode, I'm going to refer to our theme as FAP.
I probably would too. It's a long sentence where you have to put it in. I do stumble over it from time to time.
You say it.
Familial adenomatous polyposis.
There you go.
But this is because you say it over years and years and years, and you look a little bit silly when you're saying it to the professor in your department and stumble over it when you're meant to have been dealing with it for however long. But yes, this is the story of FAP, and this is a remarkable story of discovery. And look, we've actually mentioned some of the history, and when we look at the history of cancer, we have, hints, and this is in the 19th century, the hints that there is a familial linkage of cancer. And it sounds weird. It seems intuitive that you would see in history that people would just think, oh, because they had this disease, you'll have this disease because you're their parent, a child, whatever. But it wasn't obvious. And, people died of all sorts of things. So we're now living longer and we have that perspective of familial diseases that go through, autoimmune diseases, cancers and everything. So this feels inherent, but it wasn't.
We're like living historical reference textbooks, aren't we, within our family?
And look, it was, Dr. William Norris in the 19th century. Now, he was a doctor. He was seeing patients between 1820 and 1860, and he took note of this with melanomas. He recognised, started to see what we rattle off in medical school. Oh, here's the risk factors for this disease. And he just started noting that people with melanomas had this pale complexion, light-coloured hair. But he also noted that there was a family history of this, and he wrote in 1857.
It is remarkable that this gentleman's father died of a similar disease. This tumour, I have remarked, originated in a mole. My patient and his children had many moles on various parts of their bodies. These facts, together with another case that has come under my notice, would incline me to believe that this disease is hereditary.
If we go to French physician Pierre-Paul Broca in 1866, now he noticed his wife's family, had this strong history of breast cancer. And he ended up doing a genetic family tree over 60 years. He called it Family of Mrs. Z. And he found 51 of his wife's relatives of 15 developed this cancer, and he wrote about it.
A person in whom are assembled unknown conditions, which expose him to later become cancerous, can transmit to his posterity this group of conditions before they have even manifested themselves in him.
You can see they're starting to struggle with the concept, which we find probably just so simple that everyone knows about it. It's genetics and inheritance. And so they're struggling to work out, how is this person getting the disease? They're transmitting it without having the disease, but somehow it goes through their lineage. And we can, the other linkage, the reason why it was so easier for them is because these were diseases you could see. These were superficial organs that were affected. Well, what happens when it's an organ that's internal, such as colon cancer? You don't see it happening, and people end up just dying from this withering disease. And so you couldn't study it, so to speak. And the amazing thing about the story of FAP is it begins with a conversation between a pathologist and a surgeon at St Mark's Hospital in 1924. Now, the pathologist was Dr. Cuthbert Dukes, and the surgeon was Mr. J.P. Lockhart Mummery. and they discussed diseases. This disease, a rare disease of these patients, and they noted two things about it. One was these patients had multiple polyps in their bowel. I mean, they're talking about it could be up to 100 polyps in their bowel. But they also had a family history of cancer, of bowel cancer specifically. Dr. Jukes was already involved in doing research with polyps, and Mr. Lockhart Mummery then added the clinical and family history to the research that he was doing. They set up this formal system to collect data, and this was the person to do this was a lab assistant, H.J.R. Bussey, and they set up what is now known as the Polyposis Registry. They then, Jukes, Lockhart, Mummery and Bussey, collected data from 1918 to 1954, so they got backdated. So that's around 36 years of data they collected.
And straddling World War II.
Well, exactly. And what they looked at was they were learning about these tumours, these polyps. They found that they first appeared in early childhood and young adolescence, or up to early adulthood. And it was around an average of 20 years when they were 20 years old, the patients. They then noted that cancer would develop about 15 years later after the first diagnosis of the polyposis, at an average of about 35 years. There was no difference between males and females, and about 50% of children were affected. So this was an autosomal dominant disease. And they ended up collecting data of 58 families. They had 1,069 members. Of that, 218 had polyposis and 154 developed intestinal cancer. And so this is a remarkable collection over that time. They ended up publishing in 1956 the surgical treatment of familial polyposis of the colon. And this was in the British Journal of Surgery, and it was called FAP, familial adenomatous polyposis. Now, it would be decades later that we had the technology to actually find the genes, understand the genetics. And this actually comes to our guest, the Professor Sir Walter Bodmer, who was able to map the location of the adenomatous polyposis coli gene, which was on chromosome 5. Did that in 1987. The remarkable thing about this is we've learnt so much about this APC gene since then. Like, it's a large gene. There's greater than 8,000 kilobase pairs. It took another few years for the gene to be isolated and cloned. This was done by two labs in the US, in Utah and Baltimore. But we found the APC was a germline pathogenic variant. So this was transmitted from parent to child, 50%, which means that it's autosomal dominant. We know this is a tumor suppressor gene, but it's also a multitasker gene. So it controls how fast cells multiply. It has adherence to other cells and how they move. Today, we know there's greater than 1,000 APC mutations that can cause this problems.
That's a lot. This is where our guest with his statistics background plays a role.
And this is the unique linkage between cancer and genetics and statistics, and you're starting to see our understanding is still at the edges. But the amazing thing about this is 25% of FAP cases are de novo, so they're new. So it doesn't have to be just germline that this is created and it's gone on. And so there are people who are asymptomatic and then will present with colon cancer. But if they do have symptoms, we have gastrointestinal bleeding and abdominal pain and diarrhea. And what we know is essentially everyone with, 100% of people with FAP will eventually develop colon cancer. And so this is where the average is 39 years. 80% will be on the left side colon cancer. 40% of those people will have multiple cancers. And so there's even a part of extracolonic manifestations. And this people will have, 30 to 100% will have upper GI polyps as well. And There's something we don't discuss much here, but there's also Gardner syndrome, which is effectively the APC mutation outside the colon area. So these patients get desmoid tumours, sebaceous or epidermoid cysts, lipomas, and osteomas, fibromas, a whole bunch of other tumours that are due to this APC gene mutation. And look, this is why we're discussing it. It's to help recognize these people. It's helped to, put the light bulb moment that, is this a genetic, is there something genetic going on here with this patient's symptoms, or do they multiple, polyps? And so I guess, I love doing this so much because this all originated from a conversation in 1924 between a pathologist and a surgeon. Said, hey, have you noticed something unusual about this situation that's happening? And this is where this stems from.
Wonder what our conversations will lead to, Travis. Sir Walter Bodmer is a professor at the Cancer and Genetics Laboratory and Department of Oncology at Hertford College at the University of Oxford. He's studied mathematics, statistics, and has a PhD in genetics from Cambridge University, and worked at Stanford University, where he became a professor. He returned to the UK as the first professor of genetics at Oxford University. He became the Director of Research, then Director General of the Imperial Cancer Research Fund in London. Areas of interest and research include human leukocyte antigen, or HLA, population genetics, the Human Genome Project, and public engagement for science. The topic we're discussing today is Sir Bodmer's mapping of the adenomatous polyposis coli, or APC gene, responsible for the condition known as familial adenomatous polyposis, or FAP. Professor Bodmer is a Fellow of the Royal Society and was knighted in 1986. Walter is our guest on This Medical Life. Welcome.
Good evening. It's good morning to you and good evening to me.
Indeed it is. You started your career as a mathematician and statistician. How did you come to study cancer genetics?
Well, when I was at school in Manchester, which was a well-known one for anyone who could do sums and did maths well. And so they put you down for studying maths in Cambridge, and I couldn't think of anything else to study, and I liked maths, so I did. So I got a scholarship, and I did reasonably well. And so that you could do most of your maths degree in 2 years at that time. And the third year was rather like a master's, where you had a choice of subjects that you could study. Well, it so happened that in my second year, there was a stimulating overview of statistics by someone who became a world-famous statistician, David Cox. And he said, well, if you're going on into your third year like that, there's this rather well-known guy, Fisher, who gives lectures to students in their third year's maths. So maybe you'd like to have lectures from him. And he talks about mathematical aspects of genetics. Well, I hadn't a clue what genetics was. I'd learned no biology at school at all. It was a good school, but no biology. So I decided I'd better read a few books, and got hold of a textbook in genetics, well-known textbook, and one or two of Fisher's books, and spent the summer reading. And the genetics just fascinated me. It had a sort of analytical bent to it. It was something that hadn't been all solved about 100 years before. So I said, well, I think that's something I'd like to do. So I went to his lectures. Now, the term starts in October, as you know. And by the time I'd had a few weeks of his lectures, I decided I wanted to go and do my PhD with him. So I just went and asked him. Well, by that time, I'd already had the equivalent of a first-class degree in maths, and he didn't have many maths students, and he immediately said he'd take me on and sponsored me. So that actually just changed my life completely, because then I did his courses, and with his support, I did get support to start doing research under him. That was in 1953 when I'd finished my undergraduate course. So that's how I got into genetics. And statistics was because I was interested in statistics, and alongside the courses that Fisher gave on mathematical models of genetics and I first learned about DNA from him too. He gave an excellent description of it. And he was a great friend of Francis Crick's, actually. That's how I first met Francis. But there was also statistics lectures. So, in fact, I thought I might end up doing statistics, but as soon as Fisher took me on, I changed my mind on that. But statistics did play a major role in what I've done. And, amongst other things, I have at one point been a president of the Royal Statistical Society. So that's anyway how I got started in genetics. I mean, the next step was, and that was 53, 56, and then I stayed on and did, I did my 53 to 56 was my undergraduate, 56 to 59 for my PhD. And by that time, I got a junior fellowship at Clare College. But my professor then, who was the successor to Fisher, a man called Thoday, he said, well, young man, you ought to go west, maybe. Well, west meant going to learn some molecular biology. And that was very important because in those days, you remember, Jim Watson was actually in Cambridge, where he did his work with Francis Crick, and he was actually at the college where I was a student, but he left just before I came in 53. It was April 1953 when they published their famous paper. So anyway, in the end, it happened that rather with a bit of persistence, I got myself taken as a postdoctoral fellow by a very distinguished second mentor of mine, Joshua Lederberg, the Nobel Prize winner who got his prize for showing that you could do genetics with bacteria, and that was the origin of how you can do recombinant DNA technology and all that. So that's why I spent 9 years at Stanford, and that's where I started the work on HLA, which is a different story. And then I came, I married very young. I was 20 when I got married, and we had kids by the time I was 21, and my poor wife had to give up her job in economics and look after kids, but then she got a bit fed up with it, ended up working with me on HLA. But we sort of felt that, although it was marvelous at Stanford, and a wonderful place to be, that maybe in the end we'd like to go back to the old home. And so that's why we went to Oxford. And I'd thought about going. I'd seen the advertisement for the professorship, but before I'd done anything about it, I was asked, strangely for me, I was just a young kid really, how would I be interested in that? And I said yes. And so that's why I went to Oxford. Okay. So that's the sort of beginning of the story. Now, you might well say, well, what's the next thing? How on earth did you get involved in cancer? I hadn't done anything medical. My father was a doctor, and he undoubtedly influenced my interest in things human. He was Jewish, a doctor in Germany, and we had to escape from Hitler when I was 2.5 in 1938. But what happened is, of course, you got to know, even though I'd not studied medical things particularly, I got involved in human genetics quite a bit. And inevitably, as a geneticist, you got across the fact that genetics somehow had something to do with cancer. And so I was approached out of the blue about going to the ICRF, as it then was, Imperial Cancer Research Fund, to be their director of research to succeed a guy who was a good friend whom I knew very well, Sir Michael Stoker. And that just came out of the blue. I mean, they approached me in 1977, but in the end I didn't. And that was because it took time to arrange things. I went there in 1979. So that's what got me involved in cancer. And it was a steep curve because there you're dealing with a charity and you're the sort of main person. There were two people, one looking after the fundraising at that time and one after the finances, and the other one, like me, out looking after the science, which is, of course, a big part of it. It was not a grant-giving body. They had their own institute and already had 1 or 2 units that they supported in universities. So it was a steep learning curve for me because I not only had to I had to be able to talk about cancer to people as if I knew about it. So I had to learn. So I got a textbook on cancer. I learned what clinical trials were about and so on. And then I said, well, if I'm a geneticist working in a cancer institute, maybe I should do some work that's relevant to cancer, that's genetics. And by that time, of course, I'd already learned a bit more. And I was aware that the ICRF was supporting research at St. Mark's Hospital. And St. Mark's Hospital is the origin, really, of where familial adenomatous polyposis was really discovered as an inherited disease in 1925 by a guy called Lockhart Mummery, surgeon. And so at that time, having, of course, learnt about DNA and all things like that, and how you could do genetics by that time with DNA fragments and so on, the obvious thing seemed to me, here they've got a collection of families. And I'd said already in things I published, one of the ways to find out about an inherited disease, to find out where it is on the chromosome by doing just conventional linkage studies. You're using a genetic marker, a variant like a blood group, and you're using linkage, which says you're finding out ones that tend to segregate, go with the disease in the family. And so we thought, well, let's get together with the people at St. Mark's and have a look at their families with these genetic techniques. So that's how that sort of all started.
The research that you found was about 13 families, and it was 124 patients. How did you actually investigate them for this condition?
St. Mark's was a very well-known hospital, and they had a major interest in colorectal cancer. And tracing back to Lockhart Mummery and then Cuthbert Jukes, who was well known as a pathologist, Jukes' characterization of what level of colorectal cancer you have. They had, from the beginning, through Lockhart Mummery, started collecting families there. And there was a very interesting guy, Bussey, who had maintained these in a remarkable way. So an accessible collection was already there to some extent. But in addition, we managed to get in touch with people throughout the country. There was a group in Liverpool in particular, and gathered together enough families so that you've got enough that would make it plausible that you might find a linkage. So that was, I mean, it was an interesting exercise. It wasn't trivial. You had to get quite a lot of help and so on. And of course, all you did was get a blood sample from them, and from which you could make the DNA. So by that time, it was getting to be a reasonably ordinary thing to be able to do. So that's how we started that. But at that point, I had a young woman. At that time, she was young. We were all young, I guess. Vicky Murday, who had come to work at St. Mark's, but actually basically spent time working in my lab. She was a clinician learning how to be a cancer geneticist, which was quite unusual in those times. You had to get special sort of education for that. And she noticed a paper that had just come out. So we're talking about the early 1980s by this guy Herrera in the States, where he had found a person who had a variety of abnormalities, but he also had polyposis. And it turned out that he had a visible deletion in the chromosomes. Now, as you know, you can, in those days already, you had the ability to stain chromosomes, find bands, and the patterns of bands define which chromosome you were on, and you could see if you had a band that was missing. So there were three places where there were 3 bands, and one of them was missing in this patient. So that was an immediate suggestion, that maybe that was where the gene that caused that abnormality, which one knew from the family pattern would be dominant, might be based. So that gave us the clue at that time to look for markers on the fifth chromosome where that abnormality was. I should say that before, my late wife, who in the end worked a lot with me on the HLA system, she was involved, and in fact, the first study we did on the patients was to see if HLA was linked. And the reason for that is, you may know, HLA is the thing you match for transplantation to avoid as much as possible of the immune response to a foreign tissue. And she and I had been involved. We were amongst the early discoverers of the HLA system. So a simple thing to do was just to type these families for HLA. Somebody had said, mainly because it was one of the few things you could easily do, that perhaps HLA is linked, but of course it wasn't. So that's how we got into the.
Was there much excitement when you found that sort of region, or did you go, did you come with a skeptical mind, or were you coming with a, celebratory, fantastic, eureka, we found it?
First of all, the deletion that was found was just a clue. And that was still a long way from finding whether it was really where the gene was, and finding the bits of DNA that would take you close to it. So we were able, with collaborations with friends and colleagues in London and elsewhere, to pick out these segments. They're a bit like the fingerprint idea. So you had short segments of DNA that you could match to different places on the chromosome, and we looked for ones that were on chromosome 5. And we were very lucky that one of them, in the end, happened to be very close to where the gene was. So the first thing was that it showed a strong linkage. That was and then another thing turned up. There was all these things that are a bit sort of happy-go-lucky the way they happened. That we got in touch with a genetics group in Manchester, and they happened to have a patient with what seemed a very mild form of polyposis, who had a deletion in the same part of the chromosome 5. So we had access to her, and it's an interesting story because I once gave a lecture in Wales, in Chester, which I said was in Wales, which it isn't, and described this, and it was this person's mother who was in the audience, and said, "That's my daughter you were talking about." Anyway, that's another. But then that gave us access to material, which in the end enabled us to start looking for the gene. So that's the way it worked, and we were lucky that we'd found this marker close to it. We were not the people that found the gene, and that was because another group in the United States had a similar deletion, which was much smaller than the one we had. Now, the smaller the deletion is, the easier it is to get where the gene is. Sort of, in those days, it was very difficult compared to what you can do now. Now you'd have the whole thing sequenced in a few weeks and know where to look. But in those days, you had to go down step by step until you found something that might have a mutation that goes with being FAP. So we were beaten to that by the Americans, but they depended essentially on us having found where it was. But there was another aspect, because we published 2 papers at the same time, and a colleague of mine and been working with, who was at the ICIF, I brought her there from Oxford. She came from the States, Ellen Solomon. She was looking at another way you could look at these things. Now, what we're looking at is the gene defect that's inherited in the family. And a very well-known geneticist had shown, and I'd have to look his name up anyway, he'd shown that one thing you might expect, and it was an interesting idea, that if you actually needed to knock out the gene that was involved to get a cancer, if you had one mutation there already, then there are ways somatically in the development of the cancer where you might lose the other one. And so Ellen Solomon, Al Knudsen, Knud, SON, he was very well known in the States, and he came up with this idea that if you had one hit, then you'd have another hit that developed. If you inherited one hit, and the other hit, then you'd only got one thing to change, gave you the abnormality. Then the thing to look for was essentially a change in the genes there, a loss of heterozygosity. So if you've got markers that are different on the chromosome, one is with the mutation that's in the APC, and the other is not. Then if you've got the mutation with the APC and you lose the other one, then you've only got the mutation in the APC. And what Ellen showed, we're all working on this together, is that you could see in the patients that they had that loss. Right? So that was separate from the family study. That was showing in the non-familial, the so-called sporadic cases, that you had evidence that it was in the same position, that there was a genetic change that caused the disease. And that was very important, to tie what you found in the families with what you find in the cancers that are not familiar. It's a very important difference. And if you think about the well-known variations in breast cancer, for instance, like BRCA1 and BRCA2, those are only found in the families. You don't get that allelic loss. Those changes are not as important in the random cases, the non-familial case. So that was an important step at the same time. It was not just finding where the gene was, but showing that it was most probable that that gene was also involved in sporadic cancers. Now, colorectal cancers, it's now the second most killing cancer, even still in this country and more or less in the world. So that was an important step. It's an interesting lesson. Overall, you might say, well, so what? You found a gene like that, what can you do? But when you found the gene, nowadays you get the sequence. If you've got the sequence, you can find out something about it, what the function is. So finding the gene and getting the function was a big step, because that gene, the APC gene, became a very important step. And through further things, probably the first step in most colorectal cancers, maybe 50 to 80%. So it taught you a lot about what was going on as well. So it was not just. It's interesting when you think, I was very involved in the Human Genome Project, and people thought, spending all this money in this big, what's it going to do for us? Well, one of the things it's going to do for you is find out genes and what they're doing, and that you could predict would be one of the things that became possible. So that's the main way that things develop.
We're now moving into the generation of medicine, genetics, but we're also talking about bioinformatics and statistics. Do you think that more information is probably going to come from the statistics side of things, just because of the sheer amount of data that we're dealing with these days?
Well, the statistics doesn't give you anything, and I'll say that as a statistician, unless you're cell biology. Of course, statistics is important to assess the quality of the data, the way you analyze it, to see groupings of data. But to think that bioinformatics on its own can just solve things by having a lot of data, I think is mostly wrong. Now, there's a huge amount of talk about AI and that, but, for instance, I gather you're a pathologist. Well, AI, the only thing vaguely medical I have is I am a fellow of the Royal College of Pathologists by publication, not by treating patients. But, and you will know that when you have tissue sections, you do H&E staining and you see things. I very early on, I got a good pathologist who was the wife of a very distinguished human geneticist, and I would sit with her at the microscope so I could learn about what you see when you look down a microscope. And of course, those patterns are exactly the sort of thing that AI can analyze very well. So nowadays, as I'm sure you know, if you get the Journal of Pathology, it's becoming a big thing in pathology that you can give high quality advice on the analysis of a section by looking at the picture. So that's a very important contribution. But there's a lot of other sort of talk about what things can do, which I think are questionable. I mean, people would say, oh, well, you collect a lot of data on a lot of patients, and somehow you'll find something. You'll cluster them in some way by, what combinations of things they've got. AI is supervised learning. It doesn't actually help you much finding out new patterns. It helps you finding the patterns you can already predict. So I think its applications, I mean, in protein structure and things like that, in images, breast cancer imaging and so on, are very important. But also you can help the poor administrators to write 100 letters or something, and they're all about the same but with a different name. So you can do things that are going to help. But I think you really have to think about the biology and exactly what the AI can do for you. And there's a lot of hype there, in my view. I mean, I did my first computing in 1956, just before I started doing my PhD. So I've been involved in computing one way or another, so I know a bit about what goes on.
Thank you for the nuance and the context to that part of the discussion. Do you have any advice for doctors or researchers in the fields of medicine or genetics?
Well, first of all, I think science is a fascinating thing to do. And I think it's important that to try and get that point over. And science, is so important. Almost everything we do, what we're doing now, how could you do that without scientific discoveries? How would we be anywhere in treating colorectal cancer without a background to the science? So I think the first thing is to say, and that's why I got involved in public engagement of science, is really everybody they shouldn't have a professional training, but everybody should be given some opportunity to be interested in science. And some of those people should find it interesting to say, well, how can we use science to find out about things? Okay. Now, when it comes to medicine, I think there are two aspects. I'm not medical, but I've had to learn a lot as I go along. And I've had medical fellows in training in my laboratory who are enormously helpful because they create the bridge between the laboratory and the clinic, and that's very important. And I also am involved with a foundation called the Fuchs Foundation that actually provides scholarships for people who want to study medicine, having before not studied. I've got a degree in biochemistry or done a PhD in cell biology or something. And I think that's extremely important because we have to have medical people who are clinician scientists. But I would say to the people who want to go that way, you've really got to want to deal with patients. That's very important. And if you do, and you also like the laboratory work, then you can get that mixture. But as I've done, you can do an awful lot towards medical research that's fascinating without necessarily having studied medicine. You've just got to learn a lot. So I think the first thing is, are you interested in science and discovering new things? And then the other question is, are you interested in actually being a doctor as well? And you've got to answer those two questions. And I've had people, I have a lot of respect for the people who managed to mix the two together. I was for many years in the Weatherall Institute of Molecular Medicine in Oxford. Well, David Weatherall was an outstanding clinician scientist with a real expertise in the thalassemias, the sort of inherited blood abnormalities. And you need people like that, and you need to support them enough so that they don't all say, well, I don't earn enough doing that, I'm going to stick only with the clinical side.
Wow. Sir Walter Bodmer, thank you very much for being part of this medical life.
Thank you.
Dr. Tristan Rutland is an anatomical pathologist and has a subspeciality and research interest in GI pathology and asbestos-related diseases. In 2020, he was the recipient of the Conrad Muller RCPA Outstanding Teaching Award, In 2022, he received the Resident Advocate Award from the College of American Pathologists. Tristan's a regular guest on This Medical Life, and we welcome him back today. Tristan, hello.
Thank you so much for having me again.
As a pathologist, when does the consideration of FAP arise as a potential diagnosis?
That's a good question. When it comes down to polyposis syndromes or these sort of syndromes in colorectal, in colorectal and thinking about colorectal cancer, one of the things that we tend to flag and be worried about is usually the number of polyps. So a lot of these syndromes are associated with lots and lots of polyps and younger age of onset. So when we're worried about it, we're thinking if we get a biopsy or lots of biopsies from a patient that have numerous polyps, and quite often the clinicians have actually thought about this, so this is not necessarily up to us, that's where we can flag it as well. I know we're going to be talking about FAP today. There are other polyposis syndromes, which we won't be touching on, which actually might not have as many polyps. Like when we talk about FAP, and I'm sure we'll get into this, we're talking about hundreds and hundreds of polyps. But there are other polyposis syndromes that are rarer, that are still being described, that actually you only need 10 or so polyps to have them, or 10 to 20 types of polyps, and there's certain criteria. So in a nutshell, as a pathologist, we get concerned when we see lots of polyps, or we see increased amounts of polyps of a particular type.
So what is the underlying pathological cause of FAP?
I think it's a good question because it really ties into understanding a little bit of cell biology. And it's going to be a bit of a long-winded answer to that, but I think when you actually understand how it works, it actually makes a huge amount of sense. Well, at least in my mind, that's just how my mind works as well. I'm lazy. I like to remember how things work, and then if I forget them, then I can figure out, and I have to rope learn everything. But so FAP is, we have 2 copies of all our genes, and we have a gene called APC. APC, the APC. So APC is makes a protein that is essentially stops the cell from growing, okay? It actually locks onto another protein and stops it from growing as well. What happens in the body is that if you injure actually any part of your body, for example, you injure your skin, but mainly, say, your colonic mucosa, if you have a break in it as well, you've got these little attachments, the cells attached to each other. And when those attachments are broken, what happens to happen is the cells need to grow back together to complete that barrier. So the little epithelial barrier has to do as well. So normally the APC protein locks on something called beta-catenin, boom, stops it from working. And when there's a break, those little adhesion molecules aren't locked onto each other, and they send a message, and it causes that APC to break apart. And then this little protein goes off and says, right, we need to replicate until we, and they keep on growing and growing and growing until that attachment protein bangs into another one. And then it sends a signal back, and then it locks down, the APC comes back and locks down. This is a really simplistic approach, and stops the cell from growing. Okay, so it's a normal thing. All these things are normal as well. And this is what we would say is a tumor suppressor type function, for want of a better word, because you don't want APC. You want APC working. You don't want your cell to be constantly replicating as well. So we have 2 copies of that gene, as I mentioned, and people that have FAP only have 1 copy of that, which means it's still working. It just means that mathematically they're much more likely to have an error in one of those genes. And if they have an error in both of those genes, that means that cell no longer has an APC protein that can stop the cell from growing. And what happens is the cell just keeps growing and growing and growing. And it doesn't matter if it's attached to something or not. It just keeps growing and growing and growing. And then think about it like this. If you've got something that's growing and it's growing in a flat sheet, where does it go if it keeps on growing? And you get a piece of paper and you push it together, it goes up. So if you lose your APC gene because you've had you were born with one that wasn't working, then you lost another one, those cells will just keep growing, and you'll start, if that cell that lost it keeps on growing and growing and growing and actually forms a polyp. And people that have FAP have lost both copies of those genes, so hence they get these polyps. And that by itself is not cancer. The problem is that cells that keep on growing and growing and growing also, and especially when they've had one of their brakes taken off them, have an increased chance of getting more and more and more and more mutations, and that can lead to cancer. I hope that makes sense. So that's the underlying mechanism. And I would just also add that that's actually one of the mechanisms for sporadic cancers as well. Okay, so APC, loss of it, is actually one of the steps that we can get in sporadic, the 80% of other types of. So a lot of this pathway that we talk about, this stepwise approach pathway is actually this is the common way that we think sporadic cancers actually develop. And I'm sure that you've talked about this. Both of you love your history, but I'm sure that you know that they were described a FAP before they had the association with the colorectal cancer. And maybe it's because of the age, I don't know as well, but they actually sort of knew that this thing existed as well. And then it's funny, then they sort of put it all together and realized that this conventional pathway of neoplasia was typified by this syndrome they already knew existed, and that was like why we think about FAP is that it actually really highlights. It's like it's an accelerated part of that sporadic cancer. You just got one less copy of that gene, and it speeds it along to you developing tumor as well.
We've got how polyps develop, but how does that lead to cancer?
Cancer obviously doesn't happen when you have just one, or generally doesn't happen when you have one loss of one tumor suppressor gene, but it just means that those cells that are replicating more and more and more, and they have already lost one of their breaks, if you want, on their genetic sort of stability, if that makes sense as well. And anything that's growing more and more and more has an increased risk of getting more and more mutations. So you get these cells that are proliferating and proliferating and proliferating, and they're not stopping as well. They have, and then they get another mutation. For example, they can get a KRAS mutation, which is a further downstream mutation, which basically means tells the cells to even grow more and more and more, or they can lose another tumor suppressor gene like P53 as well. So it's a stepwise approach, which once again plays back into that conventional model of cancer as we understand it. You get one mutation, then another, then another, then another, then another, because it's a knock-on effect. It's like, and it is a bit mathematical. If you lose a tumor suppressor gene or the functions of something and something's proliferating, it means that the chances of you developing another one are increased, and so on and so forth. So it's a stepwise approach. So essentially, it's the first step on you developing a lot more mutations to then the endpoint, unfortunately, being invasive cancer as well from that initial proliferation of cells.
What risk do people with FAP have of developing cancer?
Okay, so FAP, full stop, just FAP, because it's a little bit more complicated because there are different types of FAP, but yeah, sort of like classical FAP, it's almost 100%. I think it is close to 100% by age 40. So everybody of getting colorectal cancer. So the chances of getting colorectal cancer are close to, if not 100% by the age of 40, which is extremely high. I should also add that there is a couple of other things as well. We've recognized as something what we call an attenuated form of FAP, which is probably a slightly different mutation. I won't go into the details of that. It means that the patients actually have less polyps. And normally when we talk about FAP, we normally say more than 100 polyps, but they're usually hundreds and hundreds of polyps. These patients tend to have less than 100 polyps as well. Same thought to be autosomal dominant loss of APC, a mutation as well. They have a lower risk. It's about 80% or so, I think, at about the 50-year mark. So they've got that, but they're still obviously very high risk as well. So classic FAP, about 100% of people get it by the age of 40, colorectal cancer. The attenuated form, which they tend to have less polyps, have a very high risk, but it's about 80% for 50. And it's very important to recognise that second group because that's the one that may very well get overlooked as well.
Does FAP affect any other organ systems, or is it just confined to the colon?
No, so that's it's a really good question because FAP is, as you would imagine, if all your cells in your body like something like an APC gene, you would think, how come you don't get, and you do get other tumours? It's just that they're less. So we just know that FAP is associated with, almost guaranteed getting colorectal cancer. but you certainly can get cancers of the other. So there are some sort of different variants, but without going into talking about Gardner's and Turncoat, yes. So your chances of getting other GI cancers are increased, but not nowhere near as much as what we would say with colorectal cancer. So yes, small bowel and gastric cancers are increased, and you can see similar polyps, so that we can see. We'll talk about the histology in a bit, but they actually look similar to the colorectal adenomas that you can get as a premalignant lesion. So you can get other things. So there's some soft tissue type things as well that you can get. So desmoid fibromatosis, which is a funny fiber. So it's a fibrous tissue. So the tissue, so it's reparative tissue, and all the tissue we hold stuff together. We can get something called fibromatosis, which is a growth of an aberrant growth of this fibrous tissue, which causes mischief because it just grows everywhere. It doesn't metastasize, but it grows everywhere as well. And that's associated with, once again, these things aren't guaranteed that you have FAP, but if you have FAP, you've got a very high chance of getting this. And in actual fact, it has the same underlying mutation again as well. And there's various other things like osteomas and such alike. So yes, it's not just confined to the colon, it's just that's the one that is obviously affected the most.
Is there any important clues in diagnosing FAP? Like when you mentioned histology, sometimes you can see something histologically that might make you think of FAP. What is that? Looking under the microscope,
you can't really tell the difference between a polyp from a patient that has FAP or sporadic type polyp that you'll get as well. They all look virtually the same, and it harks back to what I mentioned before, because the pathway is similar, or almost the same. So it's the same pathway, it's just an accelerated version of it. So when you look under the microscope, they will look like what we call tubular adenomas, tubular villus adenomas, and villus adenomas. And when I use that order, they go from something that needs a little bit of follow-up to the more nasty version, which is the villus adenomas. And those terms are descriptive terms of what they look like under the microscope. They usually have what we call low-grade dysplasia, i.e. the cells look all funny, but they're not like rampantly ugly, to high-grade dysplasia, which the cells look really, really ugly, but they still haven't invaded to then turning into invasive cancer. But essentially, under the microscope, they look the same. But it comes back to that clinical again. Lots and lots of polyps. You need to start to raise this, especially in young patients. And that's the other thing is if and we know that the polyp to cancer time frame is about 10 years. So if you're getting 100% of patients getting cancer by the age of 40, that means they're getting polyps in their 30s or 20s. This is it. Because patients can get 30. Most people with FAP will have, if they haven't been picked up, will have a colorectal cancer. And we're talking about patients in their 20s and stuff getting colorectal cancer. So getting polyps in young patients should be the clue, but it comes back to the clinical. I think that's the really important thing. Aside from the number, is there any other clinical clues for FAP? So obviously some of those other tumours, desmoid fibromatosis and such alike, I'm not sure if they once again happen in a younger patient, but I would imagine that it really does come back to, obviously family history. Sorry, that's a good point. So obviously, if you've got a strong family history of early onset colorectal cancer, absolutely, I think that's a clue that is something that you should be thinking about, and hopefully would have been picked up. But then again, there are places that might not be, and also remember that there are people out there that parents aren't their parents. For various reasons, that's always something you have to keep in mind as well. But as a general thing, outside of that as well, very good history, having lots of polyps, and young, any sort of and I know that you're going to have a really awesome clinical geneticist on later on, Amelia, and I'm sure she'll go into a lot of depth about this as well. But we do have lots of really good ways of identifying these funny sort of syndromes and identifying them and being aware of those as well. And some of them have some nuances, which I'm sure that she can go into as well. But yeah. Is any immunohistochemistry useful? Because we're talking about APC gene, and is there any expression that pick up that suggests it? No. Once again, I think it's just good if just thinking back to that model that we talked about, how it's a mutation of a gene that stops the cell from proliferating, but you lose that in pretty much all tubular, conventional adenomas, tubular adenomas and such alike. So there really isn't a way to distinguish them as well. It is what you do need to do is, once again, is genetic testing on the patient as well to find that, because once the horse is bolted, you're not going to. It's going to look the same essentially as a standard colorectal cancer as well. So yeah.
So genetics doesn't help then either. The genetics test of the polyp itself.
And this is a tricky thing, actually, to be honest with you. And this is something that Amelia, we don't tend to do genetic tests of the polyps. We do genetic testing for cancers, and then the next step is, after they get referred to a specialist, they will do actual testing of the patient's DNA, which is really quite complicated. The only thing I'd add to that is and once again, I'm sure that Amelia will expand upon this, is that with cancers, you're going to be very careful where we do cancers, because if you've got an underlying syndromic or underlying germline mutation, the cancer could maybe so crazily mutated that might have actually been wiped out. So cancers like to wipe out, sections of chromosomes and stuff like that. So it's not always 100% reliable, but because it's really good that you're talking about, because I'm sure she'll be able to sort of articulate that a lot better than I can as well. But so as the answer is no, not for polyps, no.
So looking at the prognosis then for patients with FAP, knowing that they have cancer around the age of 40.
Even younger than that, yeah, sorry. I'm just saying that's the upper limit, so just by that. But yeah, but we're even talking about patients in their 20s as well, so yeah.
So if they then have surgery to remove their colon, what's their prognosis like?
So that's a really good point as well. So what happens? Well, if you obviously remove the colon, the chances of you getting colorectal cancer plummet. But on the flip side of it, I did mention that you also have a risk of getting other cancers, though they are lower. So it would be very interesting to find out what the actual guidelines for screening or surveillance. It would actually be surveillance. You're not screening now. You're actually moving into the surveillance territory as well for upper GI cancers would be as well. But I would, because these patients do have a high risk of getting small bowel type cancers and gastric cancers as well, they would need obviously be surveilled in the upper GI sense to make sure that they're not got any sort of funny polyps and so on and so forth as well. They need to obviously be aware that they're not going to get a second cancer, because obviously they've got this predisposition to getting tumours. And obviously if you remove their whole colon, well, the chances of them having colorectal cancer, if there's nothing in that colon already, is but the chances of them getting a secondary malignancy from the underlying condition is obviously still there as well. You can't remove the whole digestive system, so therefore it's important to make sure that they're getting adequate follow-up as well. And like I said, there are other things, and there are like I said, I mentioned it, there are other variants of this particular thing that you can get brain lesions and brain tumors and so on and so forth as well, and it really is tailored to what they're thought to have and what particular guidance. And that's really moving into quite some specialist sort of areas there. But they do obviously but the principle of it is that even if you move the patient's colon, they still have a risk of cancer in another organ system, because the genes have got this predisposition to turn into something nasty if they get a second hit, because of the susceptibility as well, if that makes sense.
Tristan, as always, we appreciate the candid and reflective nature of the way you explore the topics we put in front of you. In closing off today's discussion, do you have any advice for doctors who have patients with FAP?
I suppose we've just sort of covered most of it, is that obviously young patients that have lots and lots of polyps or a very strong family history of colorectal cancer. And I will just preface that most people that have a family history of colorectal cancer don't have FAP or one of those syndromes, but if they have especially young, they should always be thought about. But I suppose one of the ones is, I just did want to bring up that there are some other forms of polyposis syndromes. There's the attenuated form of FAP, so they don't have to have the hundreds of polyps, less than 100 polyps, but that's something that should be aware. So there are, just because patients don't have hundreds of polyps, they just, it should be thought of. And if they're getting, and this is going away from FAP, but we're still talking about this polyp, if they're getting lots of funny types of polyps as well, like, sessile serrated adenomas or lesions, even lots and lots of hyperplastic polyps in weird places, they should probably just be considered to have some sort of screening about these. These things our understanding of FAP And all these polyposis syndromes is expanding as we understand the underlying molecular. But it really comes back to that common sense type thing. If patients have lots of polyps and they've got a family history of colorectal cancer, yeah, they should be looked into further. But if they start to get funny types of other polyps as well, maybe it's just worthwhile just looking a little bit further into that to make sure they don't have some sort of underlying syndrome that that is now that we're starting to describe as well. So, but yeah, I think it's just thinking about that and being aware that there are attenuated forms of these ones which can be tricky to pick up because they present later, and they might not have the classical hundreds and hundreds of polyps as well. And the other thing is, if you do have FAP, don't think it's just a colorectal thing. There are other tumours that can arise as well in the GI tract and outside of the GI tract that can also obviously affect the patient as well.
Goodness me. If it was too easy and straightforward, we wouldn't need doctors, would we? Dr. Tristan Rutland, thank you once again for joining This Medical Life.
Thank you so much for asking me.
Dr. Amelia Ip trained as a medical oncologist before subspecialising in cancer genetics. She's a staff specialist in cancer genetics at Liverpool Hospital, which services southwest Sydney, and co-chair of the EviQ Adult Cancer Genetics Reference Committee. Amelia is our guest on This Medical Life podcast. Welcome.
Thank you very much.
What is the actual genetics, the pathology of FAP?
So, familial adenomatous polyposis is a clinical syndrome where the most characteristic features are the development of hundreds or thousands of colonic polyps and a high risk of developing colorectal cancer. And it's responsible for around 1% of colorectal cancer diagnoses. So FAP is autosomal dominantly inherited and in the majority of cases is inherited from one or other parent. However, for 15 to 20% of individuals, it is a de novo pathogenic variant that causes the syndrome. And so there is no significant cancer or polyposis history in the family. Correlations have been made between the position of the variant within the gene and phenotype. This means that variants in some parts of the gene may be more likely to cause particular manifestations compared with other parts of the gene.
So when we're looking at it, what are the health risks of having FAP?
In classic FAP, the polyps can begin to develop from childhood, and this is the reason that annual surveillance is recommended to commence between 10 and 12 years of age. The surveillance may take the form of colonoscopy, or in children, flexible sigmoidoscopy may be considered until polyps develop, from which time full colonoscopy is required. In families with an attenuated form of FAP, where polyps develop later and are less profuse, annual colonoscopy is recommended commencing between 18 and 20 years. Once the polyp burden becomes too high to safely resect, colectomy is recommended. Depending on the surgical approach, surveillance of residual bowel is required after colectomy. So while colorectal polyps in cancer are the most prominent feature of FAP, there are a number of other associations to be aware of. Upper gastrointestinal tract polyps in the stomach, duodenum and small bowel are almost universal. They are mostly benign but require surveillance because of an increased risk for cancer. Duodenal and periampullary cancer occurs in around 15 to 20% of individuals. Gastric fundic gland polyps are very common in FAP and may be present in children, but again, these are mostly benign. The presence of large polyps or carpenting of polyps is a risk factor for gastric cancer. So gastroduodenoscopy for asymptomatic individuals is recommended from age 25 years, with frequency every 1 to 3 years depending on endoscopic findings. It should be performed by an experienced endoscopist, though, with detailed understanding of FAP. Another manifestation of FAP is called desmoid type fibromatosis, and this particularly affects the intra-abdominal cavity or the abdominal wall. A germline APC pathogenic variant is found in around 12% of individuals with desmoid tumour in these locations. but only around a third of individuals with FAP will develop desmoids. So desmoids are benign tumours but may cause significant morbidity due to obstruction. No routine surveillance is recommended, but investigations should be initiated with any concerning abdominal symptoms. Other cancers that have been associated with FAP include thyroid cancer, hepatoblastoma, and medulloblastoma. Thyroid cancer may occur in up to 10% of individuals with FAP, and most commonly in early adulthood, between the mid-20s to mid-40s. An otherwise very rare subtype called cribriform morula variant of papillary thyroid carcinoma is enriched in individuals with FAP. Current Australian guidelines recommend baseline thyroid ultrasound at age 18 years and 2 to 5 yearly, depending on findings. Hepatoblastoma may occur in up to 2% of children with FAP, with a median age at diagnosis being 18 months. There is no evidence of benefit for surveillance for hepatoblastoma. Less than 5% of medulloblastomas, a predominantly childhood tumour, are due to FAP. Those that are diagnosed in FAP individuals are of a specific WNT subtype of medulloblastoma and appear to have a good prognosis with appropriate treatment. Although higher than the general population risk, the absolute risk of developing medulloblastoma for an individual with FAP remains low and is under 1%. There is therefore no evidence for surveillance for medulloblastoma in asymptomatic individuals. Other non-cancer manifestations of FAP that should be mentioned include multiple osteomas of the skull or mandible, retinal lesions called congenital hypertrophy of the retinal pigment epithelium or CHERPI that are both multiple and bilateral, and the presence of epidermoid cysts.
Why do patients with FAP essentially have a 100% risk of developing colorectal cancer?
It's true that untreated individuals with FAP have a very high chance of developing colorectal cancer. This is because of the type of polyp that predominates. The vast majority of polyps are adenomas. Not all adenomas will acquire the additional mutations that allow them to progress through from low to high-grade dysplasia and then into invasive cancer. However, a high burden of polyps in the bowel for FAP individuals increases the likelihood that one or more polyps may become cancerous. Whilst polyps may begin during young adulthood, the average age of colorectal cancer diagnosis is in the late 30s, but 10% will develop colorectal cancer by the age of 30 years, highlighting the reason for early surveillance. The endoscopic surveillance and then surgical preventative approach mentioned earlier has been shown to reduce both colorectal incidence and mortality for individuals with FAP.
Now, you've already touched on this, but what are some of the other health implications of patients with FAP?
Yes, we've discussed the medical consequences of FAP, but it's important to acknowledge significant psychosocial impact of FAP for individuals and families. The diagnosis of FAP comes along with a number of proven strategies to reduce cancer-related mortality, but this includes multiple medical appointments and invasive interventions which often commence in childhood. When optimally managed, that is, with colectomy appropriately occurring once polyp burden becomes problematic but before cancer occurs, individuals are left with altered bowel function. Left untreated, it's life-threatening. So the response of individuals to the diagnosis of FAP for themselves or a close family member will vary and may depend on lived personal and family experience, age at which the FAP is diagnosed, acknowledging that predictive testing may occur during formative childhood, adolescent, or young adult years, coping strategies, illness perception, and support networks. There may be expressions of anxiety, fear, or guilt for oneself and for children. On the other hand, identifying the hereditary predisposition syndrome that has affected health outcomes in a family can provide a sense of empowerment, especially when effective risk-reducing strategies are available. Supporting the psychosocial well-being of FAP families is a critical component of their care.
So what are some of the common questions you get asked by patients with FAP?
The information requirements vary widely between patients. When there's a history of polyposis known in the family, relatives may have quite a good knowledge of FAP, the surveillance and future surgical implications, especially when it's a parent or siblings that were affected, as it has become part of their lived experience. In these situations, patients may have very few questions beyond wanting to have the genetic test done and to find out whether or not they have FAP. This is not always the case, and so sometimes even if children are aware of bowel-related issues and surgery, they may not have detailed understanding of the underlying genetic reason for this, especially if polyposis has been successfully managed with colectomy and no cancer was ever diagnosed in the family. In this situation, we need to go back to the beginning, explain genetic testing that has happened in the family, what FAP is, potential implications of having FAP with regards to polyposis risk, extracolonic features, and risk management. For parents, though, the most common question may be when to bring their children in for predictive testing. This will depend on the phenotype within the family. In general, when there is a classic phenotype, children are ideally tested by age 10, and when there's an attenuated phenotype, testing is recommended to happen by age 18 years.
So how important is familial testing?
The phenotype for classic FAP is strong, and in the past the clinical findings of polyps in a relative was considered enough to diagnose FAP. But these days we would advocate that identification of carrier relatives through predictive genetic testing for the known familial pathogenic variant is preferable to clinical assessment. Genetic testing can be undertaken on a simple blood test, and it is definitive, as the relative either has inherited the specific variant or not. Individuals who have inherited the familial APC variant can commence surveillance, whereas those without the familial APC variant can avoid invasive and costly interventions. Additionally, if a relative who is considering starting a family can confirm their genetic status, they have the option, although not obligatory of course, to consider pre-implantation genetic diagnosis as a way of trying to minimise the chances that the APC gene fault will be passed on to their future children.
What should flag to doctors that this could be a familial cancer syndrome?
The presence of multiple colonic polyps is the clearest flag to consider FAP, especially if they arise during childhood or young adulthood. If an individual has between 20 and 100 adenomatous polyps, there is a 10% chance of finding a heritable fault in the APC gene. This rises to around 60% if there are between 100 and 1,000 adenomatous polyps, and 80% if over 1,000 of these types of polyps are identified. Other flags include the presence of other unusual clinical features that are associated with FAP. So just to summarise, the presence of intra-abdominal or abdominal wall desmoid tumours in a young adult, the presence of congenital hypertrophy of the retinal pigment epithelium when bilateral and multifocal, the diagnosis of hepatoblastoma under the age of 5 years, medulloblastoma diagnosed under 18 years of age with the WNT molecular subtype, or carpeting of the gastric wall with polyps.
So when should cancer geneticists be contacted?
So EviQ is a New South Wales Cancer Institute supported group that has developed referral guidelines for colorectal cancer or polyposis related conditions. These guidelines are available through the EviQ website. Relevant for adenomatous polyposis, the key recommendations are to refer any individual with a cumulative count of between 10 and 20 adenomas under the age of 60, or 20 or more adenomas at any age where an intra-abdominal or abdominal wall desmoid tumour is identified in an individual under the age of 60, where there are numerous gastric fundic gland polyps, or of course when a person is a relative of an individual with known FAP to discuss predictive genetic testing. Another situation would be when individuals were diagnosed with FAP or had a colectomy due to polyposis perhaps a decade or more ago. In this situation, it's useful to ask whether they have had genetic testing, especially if you have no record of their genetic test report on file. If they only had a clinical diagnosis, then genetic referral can be important to confirm whether there is an identifiable hereditary gene fault in the family or not. And this is useful, as mentioned before, for predictive genetic testing of family members down the track. The referral can also be useful to provide an update on extracolonic risk factors, their management and surveillance guidelines.
Amelia, this has been fascinating listening to this and the dense complexity of this field. I do want to ask, though, what attracted you to this field?
I came through a medical oncology background, and towards the end of my training, there was the opportunity to do extra work and training in cancer genetics. I applied for a fellowship training grant in cancer genetics through the New South Wales Cancer Institute, which was for 2 years. And this allowed me to really do a deep dive into this area. So once I came out at the other end of that, 2 years later, I have just stayed with cancer genetics. It's a fascinating area. We get to speak to many people and families. I love the counselling aspect of it. I love the incredible pace of research that's occurring in the genetic sphere, and the fact that you can really make a difference to the lives of people and families to help them change perhaps the future outcome, managing a condition which they may not have, they may have an experience of a lot of cancer in the family. Knowing that there's a gene fault can change outcomes for the future with the effective risk management that they can access.
And I'm sure with the pace of change, we're going to have to keep updating this episode every few years. Just finally, do you have any thoughts or advice about clinical genetics in colorectal cancer that's associated with polyposis?
Yes, of note, not everyone with several hundreds of polyps will be identified to have a heritable fault in the APC gene, which we've focused on today. This could be because there's a fault in the APC gene that's hard to detect or classify with current technology. For example, a variant in an intron or a regulatory region, or due to a whole gene deletion that may not be detected through some testing pipelines. Somatic mosaicism has also been documented for FAP. This means that the APC gene fault is introduced in the early post-conception period and affects some, but not all cell lineages. If, for example, the APC gene fault is present in part or all of the bowel wall, but not in the blood, you may find that the blood test for APC does not reveal any pathogenic variants or only at low frequency, whereas testing of the APC gene in tissue reveals the same APC gene fault from multiple bowel wall sites and polyps. There are also other genes that have been linked with polyposis. Although it's useful to be aware that there are other genes that have been identified, a detailed knowledge of these very rare genes is not necessary for most general practitioners, and they will be assessed as necessary by the Cancer or Clinical Genetics Service.
Dr. Amelia Epp, thank you very much for joining This Medical Life.
And thank you for having me.
This Medical Life is recorded in the Talked About Marketing Studios in Adelaide. For show notes and more information about the podcast, visit thismedicallife.com.au. You can contact the hosts on social media. Dr. Travis Brown can be found on X. His username is at Dr. Travis Brown. That's Dr. Travis Brown. And Steve Davis can be found on LinkedIn. Go to linkedin.com/in/The Real Steve Davis. This has been a PathNotes Proprietary Limited production.
Are you a podcaster? Maybe you've got that big idea and you're looking for a network to join. The multi-award winning Ozcast Network can get your content to eyes and ears all over the world. Join now for the first month free, and you could be featuring this sound at the beginning of your podcast.
Ozcast.
Simply head to ozcastnetwork.com for details.